Hi everyone and welcome to the webinar today. Today's webinar is entitled T-SNE Transformation and Image Cytometry Aids in the Study of Emerging Model Organisms, and it's presented by Andrew Box from the Stowers Institute for Medical Research. The webinar is based on research presented by Andrew at the CITO 2017 meeting in Boston. The team at Novo Software is excited to be hosting the webinar today for Andrew to present his work as it combines many of the new and powerful features that FCS Express 6 offers, as well as utilizing the powerful imaging flow cytometry methods and techniques available from Amnis instruments. We're also pleased that the team from Amnis Millipore Sigma can join us on the line today to answer any Amnis-related technical questions that may arise. As the webinar proceeds, please type any questions you may have for Andrew, Novo Software, or Amnis, and we'll follow up during and at the end of the webinar. Before we started today, we wanted to outline some of the key points for working with Amnis-derived data files in FCS Express. And due to the collaboration between Amnis and Novo Software, direct compatibility between ideas-derived .daf and .cif files has been enabled in FCS Express. So that means that FCS Express may be used to work directly with those file types with full access and linking between raw data points, plots, and images within FCS Express. That being said, FCS Express in no way replaces Ideas software, but it acts to complement it by allowing data analysis and a Microsoft Office-like interface, which is easy to use and learn, and provides high-quality visualizations, plots, and reports that export directly to the format of your choice. Further integrated tools in FCS Express allow for easy-to-use data transformations to perform t-SNE, v-sne, clustering, PCA, and SPADE, as well as direct integration with R for any custom transformation and scripting needs. Andrew Box and his team have utilized most of the functionality described, and we're pleased to give him the floor today to present his work on t-SNE transformation and image cytometry aids in the study of emerging model organisms. So with that, I'm going to turn the presentation over to Andrew.
All right, thanks, Sean.
Yeah, thanks again for presenting today, Andrew, and we'll let you take it away.
All right, thanks a lot. So again, thanks to everybody for letting me come and have a chance to show you guys some of the work that we've done with our image stream and then using some of these new features in FCS Express. I hope I can show you something interesting and potentially useful. Hide my control panel there. Okay, so I'm going to jump right into it. If anybody hasn't used or seen an Amnis, it's an imaging flow system. So, you know, instead of a pulse of fluorescence for every cell that you analyze, you actually get an image. And it's a fluorescent system, so, you know, it can be configured with, you know, up to, I think, 5 different lasers and a bunch of different detectors. So it's really like a multicolor flow system that can take images. And, you know, the highest resolution you can achieve is with a 60x 0.9 NA. So you get images that are, you know, similar quality to, you know, a decent wide field scope. And so we found this to be really useful in some of the work we've been doing with organisms and tissues that are, you know, sort of non-mouse, not established model systems. So, you know, we have a situation where commercial antibodies are not really available, and, you know, the community for these model systems is kind of in the process of building up a reagent set to be used. And so what we've done is sort of leverage the image system to take advantage of just some cell-intrinsic features, because with bright field, side scatter, you kind of get those for free, right? And so you can extract a lot of things from the cells that tell you a little bit about their morphology and potentially their cell type. And so we have here really a multidimensional data set, because from every channel that you collect, you can get a number of different feature values that, you know, inform as to, you know, parameters related to cell size and shape and texture and all these different things. And so it's really best treated as a sort of a high dimensionality data. And so quickly, I'm going to describe kind of 2 little short stories today in terms of the science that we've done. And one of them is in this interesting organism, Osteonyx mexicanus, and it's a Mexican tetrafish. And one of our investigators here at the Stowers Institute, Nick Roner, his group is working with this fish and using it as a model for sort of evolutionary biology, because what's interesting is that these fish live in rivers down in sort of north central Mexico. And the ancestral form is the surface version, and it looks pretty much like a normal fish, right? It's got eyes and pigmentation. And there's several caves that are nearby to these rivers where the surface fish is found. And over time, what's happened is, multiple times the surface fish have washed into caves. And then because of the really drastically different selective pressures that are present in the cave environment, they've evolved a cave-adapted phenotype. And so this is actually, given rise to parallel evolution, because not only has this occurred in one cave, it's occurred at least 2 or 3 times, separately in several nearby caves. And what's interesting about the cave-adapted phenotype is that, they're blind fish. So you can see this fish on the right doesn't have eyes. They've lost their pigmentation. And then they have a very high body fat, high blood glucose, fatty liver phenotype. And so this is very reminiscent of sort of a late stage type of diabetes. And so it's actually been used recently by Nick and his group as a model for studying diabetes in mammals. And what's very curious is that the cavefish, despite having some of these physiological features that are very much like diabetes, is that they live a normal lifespan and that they're otherwise seemingly very healthy, and they don't have a lot of inflammation in various tissues, including liver. And so the question is, sort of what is happening with these guys in terms of genetics and physiology that allows them to be sort of resistant to this diabetes-like phenotype and still maintain a sort of normal physiology and health and lifespan. And so they're looking at several things, obviously, sort of insulin resistance, insulin signaling, and metabolic physiology is one area of their work. But separately, this project is something I've done with Rob Poist, who's a postdoc in their lab. And we're really interested to try to understand what is happening with respect to the sort of host parasite or parasite pathogen and host immune system interactions, because we can hypothesize and demonstrate just from sampling the different sites that the complement of parasites and pathogens that the fish encounter in the surface rivers is really drastically different from what they encounter in the caves. And it's known that the immune system and pathogens tend to be involved in sort of an evolutionary battle. So we'd like to understand what changes, if any, are occurring in the immune system that help these fish be resistant to diabetes-like phenotype, and also just in general give rise to their sort of enhanced adaptive capability to live in the rivers or in the caves. Oops. So a couple of, we kind of distilled this into a couple of simple questions just to get started that we thought we could answer using image cytometry. First, can we detect just simple differences in the frequency of different hematopoietic cell types in surface fish versus cave fish? And this is looking in head kidney tissue, because in fish, the hematopoietic tissue is head kidney. They don't have a hematopoietic bone marrow. And second, can we see differences in the capacity of these different immune cells to respond to antigen stimulation? So in our case, we've been using LPS, bacterial cell wall antigen, to stimulate and activate the immune cells, and then culture them, and then look with the image cytometer and ask, can we see differences in sort of the cells that are expanding in frequency or changing morphology, et cetera. So here's an image panel of one of the surface fish head kidney samples. And so we can see a bunch of different cell types here. We've collected bright field, side scatter. We stain DNA with DRAC5 so that we can assess things like nuclear morphology and nuclear to cytoplasmic ratios. And you can see right away, there's really a number of distinct different cell types here in terms of their morphology. But we don't have at all commercially available antibodies. We're currently looking at trying to find some cross-reactive antibodies for other species, but so far haven't had a lot of luck. But nevertheless, we've got a lot of information content just right here without even using antibodies. And in fact, there's enough, there's a large enough number of features that it's kind of difficult to just go through by hand and look at every feature versus every other feature and gate by hand and be confident that you haven't either sort of biased your analysis or missed some interesting population. By the time you look at a couple dozen features, it's really hard to look at everything. So how can we, sort of distill some of this high dimensionality data down? So we started by just looking at dimensionality reduction techniques. And, many of you, I'm sure, have seen either PCA or tSNE used in sort of other situations, whether it's with CyTOF data or even single-cell RNA-seq data, any case where you have sort of a large number of parameters by some number of samples. So this is just sort of an illustration of, how tSNE can sometimes perform better than PCA in terms of resolving unique populations. It really just depends on what the shape of the underlying data is, how well it works. But I think this is just a really nice example that shows that tSNE can be superior, at least in resolving different discrete populations from a high dimensionality data set. And this Amnis data set is a test data set that's used a lot in machine learning, and it's just a huge set of several thousand hand-drawn numbers. And then, PCA or tSNE here is operating really on the raw pixel matrices, and you can see that tSNE has done a pretty nice job of forming, pretty discrete populations of all the different hand-drawn numbers. It's got some downsides, because it's, it's stochastic. There's the final solution. You don't get an equation out like you do with PCA. So PCA can be applied to new data, and the sort of location on the PCA space is meaningful in terms of how it maps back to high dimensionality data. tSNE is less so, meaning that if two events are close together in tSNE space, they're more similar, but you can't really say much about, one event that's far away from another versus some other event that's far away from another. But nevertheless, it's very useful because you can identify discrete populations. So that's sort of basically what it is. And then just quickly, I'll say that we also use some clustering, because what tSNE or PCA doesn't do is assign an object or a cell to a group. So tSNE is very useful for data exploration. You can sort of have a look. Do you have some cell types or events that are, clustering together in a population? You can get into FCS Express and gate on those directly, just using DAF files, and make some data grids with images to actually have a look at them and drag your gate around and explore the data. And so it's very useful for display purposes and data exploration. But if you want to do sort of a unsupervised analysis that allows you to do some quantitation, without any type of manual gating, we went ahead and decided to go a step further and perform clustering for that purpose. So the clustering algorithm that we settled on is called Xshift, and this came out of the Nolan lab. This paper that I referenced here is the paper that describes kind of how all this works. It's a k nearest neighbor density estimation algorithm. The environment for this is called Vortex, and it's free and available on GitHub. And so this operates on FCS files. So this allows us to assign every cell in a huge data set from a bunch of different samples into a cluster ID, and then start doing some statistics and sort of unsupervised calling of which clusters are different between a treatment group and a control, or one animal type and another, for example. So this clustering, combined with some dimensionality reduction techniques, kind of comprises the more holistic view of the whole workflow. And just an overview of kind of what is this workflow looking like. Start by getting your samples. In our case, for this mini story, it's the head kidney cells from cave fish or surface fish. And we really have several sort of groups here, several conditions. We either have surface fish or one of two different cave isolates. And then for each of those, we cultured 24 hours either with or without LPS treatment. And then we just harvest those cells, stain them with DRAC5 to label DNA, run them on the image stream, get bright field, side scatter, and DNA channel, and then go straight into ideas. Like Sean mentioned, FCS Express won't replace ideas, because you still got to go to ideas and do your, feature extraction. Normally we're generating features in addition to the default features and setting all that up in ideas. I usually gate out, dead cells or huge aggregates or things that I know I don't want to analyze downstream. You can do that in ideas. And then I export FCS files, which will feed into the clustering environment. And then you can also, or go straight into FCS Express and run tSNE to do some sort of preliminary exploration of the data. Or if you get, tight enough cell clusters there, that can be a good endpoint, because then you can gate and quantify. So what's really nice is that for when I do the clustering in Xshift, this is operating on FCS files, right? So what I'd really like to do is get cells assigned to clusters and then go back and be able to look at the images for those cells and try to understand, what do these look like, and do they have a morphology I can identify or use to build hypotheses. And to do that, we've used the R integration in FCS Express. So Xshift will give me a big table out, basically cell number, and then cluster ID and some other things, and I can use R integration and FCS Express to put that all back together, and then open DAF files, and then gate on cluster IDs, for example, or make some plots of cluster IDs for different samples. And I'm happy to share, some code and advice and experience with this after the talk. Sean will share my email. All right, so what does this look like? So here I'm just taking DAF files, opening those directly in FCS Express after I've set up all my features and ideas, and then running tSNE on. Here I've settled on 6 different feature values, just kind of as an example. And the bottom 3 plots are colored by frequency. So we've got tSNE X versus tSNE Y for a surface fish, or the Tanaha cavefish or the Pachon cavefish. And these are merged, a bunch of samples and a bunch of replicates. And what you can, a couple of things you see right away is that there's a bunch of, population density regions that, we would expect would correspond to cells with sort of similar morphologies in terms of their combination of feature values. And also that the frequencies of these different populations do differ between the cavefish and the surface fish. So we're sort of learning something right away just from looking at these plots. And then in the top right, I can take the same type of density plots and now color them by feature intensity value. So for example, the very top left plot in the upper panel is colored by side scatter intensity. So, the areas of red density are high side scatter and areas of blue and black are low side scatter. Very top right, colored by area of the bright field mask. So the red cells, these are the larger objects, and blue and black are the smaller ones. So, it's useful and informative to lay things out this way, and then run tSNE maybe on several combinations of feature values features that you think will have, information content. And, it's a nice way to sort of do this iteratively, start by including a bunch. If you see that the plots colored by feature value don't really contain much information, they're sort of just all blue across the field, sometimes it performs better just to take those out because essentially you're just adding noise. And so we can also gate on these. So what I've done here is gated several populations that appear to be differing in frequency and abundance between the different fish types. And so once you've gated regions in tSNE maps, you can then make data grids and show the cell images. And these channels come from whatever you've set up in ideas in your DAF file. So all your image settings and overlays and stuff, you can just say, okay, show me the overlay I set up. So here I'm showing just bright field track 5 overlay images. And we can see several interesting things. So population 1 is a highly granular. This has a higher side scatter. So these likely correspond to like a myelomonocyte population. We can kind of confirm that by looking back at area versus side scatter, and they're definitely in the location where we know myelomonocytes should be in fish. Populations 2 and 3 are more lymphocyte-like, especially population 3. They're smaller, round cell, lower side scatter, high nuclear to cytoplasmic ratio. Population 4 is something that, it's not totally clear what that is. It has slightly different morphology, some little sort of small projection type of things. Populations 5 and 6 are enriched for red blood cells. So fish, like avians and reptiles have a nucleated red cell. They're an oval type of larger cell, so they're very distinct. You always know when you see a red cell in these. And then you can see at the bottom left, I've just made a table to show me frequency of these across the different fish types. And so we can see already, okay, yeah, we've got several morphologies here that we can identify consistently, and they do differ in frequency just in the untreated, head kidney samples from the different animals. So then if we do Xshift clustering on the feature values in Xshift and make spanning trees in the Xshift package, we can, and this is just a spanning tree is basically drawing out the clusters, each cluster connected to its most similar nearest neighbor cluster. Basically the same idea as if you've run SPADE or something like that. And then, we can have a look at these branches and go into FCS Express and gate on, combinations of clusters that form a branch, for example. And when I do that, we can see that, this sort of makes some sense. So the sort of upper left branches are all very lymphoid-like in their morphology. It's a little bit heterogeneous, but definitely these are where most of the smaller round lymphoid-looking cells seem to be. This sort of middle branch that I've labeled myelomonocytes, this is where essentially all of the higher side scatter cells are. On the bottom, the sort of entire bottom right of this tree, these are all sort of different flavors of red cells. And some of this could be from just orientation as they run through the flow cell. But interestingly, the cluster labeled RBC progenitor ? That sits sort of at the base of all those clearly differentiated red cells does have kind of an intermediate morphology. So we can start to sort of build hypotheses and then think about how to go back to the bench and test whether in fact maybe that population is progenitor or something that's sort of intermediate between a non-red cell and becoming a mature red cell. Okay, so now I'm working with a slightly different data type. So what this plot is generated from is from the Xshift clustering environment called Vortex. Once you run all the clustering, you can make a table. And the table is every row is a cluster, and then the columns are counts of cells in that cluster by sample. And so I can run this Mds plot in R on that, and that's very similar to PCA. It's a sort of combinations of higher parameter data. And I did this because I wanted to see, can we detect sort of the two primary variables in this experiment? If the information content in our counts of cells per cluster by sample, actually does contain information that's correlated to the conditions we have, then we hopefully should detect that. And it looks like we can. The surface fish or samples are all on the left side of the x-axis. And then if you look at each pair of samples, meaning each fish type, you can see that no LPS is lower on the y-axis and its mate pair, which is higher on the y-axis. And so it seems like y-axis is capturing more of the LPS treatment effect, and then x-axis is capturing sort of fish type, surface versus K-fish. So this was encouraging and allowed me to think it was probably worth going on and doing some more statistics. So again, with that same data type, which is just counts per cluster by sample type, we can do some statistics. In this case, I've done negative binomial regression. So same type of statistical modeling you would do to test for significance in RNA-seq data, because the data shape is the same, and it's a good way to, test for significance and also consider the fact that clusters with lower counts are likely to have more, less certainty essentially, rather than just looking at percentages of cells and clusters and then calculating a p-value from a t-test. This actually allows you to have more confidence in smaller changes when you've got a cluster with lots of counts, for example. And so when I set it up, and I do this in R, and I set this up, and I can run tests and say, okay, on the far left, in Pachone fish, LPS versus no LPS, show me the clusters below some p-value and tell me what they are. And similar to surface fish, and then or Tanaha K-fish versus surface fish. So now we can actually get some without any supervised input or gating or anything. We can actually get some statistical output that tells us clusters that are likely to be meaningfully different between different treatment groups. Okay, and I should just say at this point, because I don't think I show it, but what you can then do, of course, is go to FCS Express and say, all right, now let me look at these clusters, set up some figures, have a look at what these morphologies look like, and lay out some nice panels like that, and then kind of build that into your biology workflow and hypothesis building. And so this is something that would be very difficult to do without the FCS integration of all this stuff, because getting cluster IDs back into ideas and then some of these other, and working with lots of samples, all together is a little bit harder in ideas. And so it's been very useful to be able to do that in FCS Express. So just to wrap up that first little story, essentially, yes, we can detect differences in frequencies of different immune cell types based on morphology, and it's just sort of using no antibodies in the head kidney tissue from K-fish versus surface fish. And second, it does look like there's a differential capacity of these different populations to respond to LPS stimulation. So we really need to follow that up, go in and have a closer look at what those cell types are, and then maybe sort of build in some other assays to try and understand, biologically and mechanistically what's occurring there. And so next steps include, I'll back up a little bit. Our overall goal is really to try and build sort of a cell type ontology, especially in some of these systems where, they're not animals or tissues that have been studied for decades. And so, we don't really have a full hematopoietic lineage tree, for example. So we like to build up an ontology, for different cell types, and includes morphological features. And so we sort of have that from this. We can say, all right, cluster 17 is defined by these morphological traits in terms of size and granularity and these other things. And then start building onto that some things like functional data. So, we can do like a in vitro or in vivo phagocytosis assay, and then run this same type of analysis and overlay sort of a phagocytosis positive readout. Say, okay, which of these populations are phagocytosis competent? Do some cell sorting, and then ask, are any of these populations capable of engraftment if we do sort of not bone marrow, but head kidney, hematopoietic cell depletion in an animal, and try to rescue that. And similarly, like single cell RNA-seq, or to try and understand where can we overlay some gene expression matrices onto some of the populations we see by morphology. Because obviously the morphology data by itself is not going to capture differences. T cells and B cells both pretty much look the same by this assay, but I think we can sort of dissect that out a little bit more with other approaches. All right, so this is sort of a shorter second story, just to give another example of where we're using a similar type of workflow. And in this case, we're working in planaria. It's a type of flatworm. So Alejandro Sanchez Alvarado's lab at the Stowers Institute is working heavily in this organism and using it as a model for adult stem cell function and regeneration, because these flatworms have a very amazing capacity to regenerate themselves if they're injured or damaged. So you can actually take these in the lab and chop them up with a razor blade into little tiny pieces, and each piece can give rise to a completely new animal. So, obviously there's a lot of different tissue types in these animals, even though just by eye they don't look like much. They've got musculature and nervous system and epithelia and gut, pharynx, eye spots, and somehow a little like fragment 1/30th the size of the animal just from the tail can within a couple of weeks grow back an entire animal. So it's a great model to try to understand sort of stem cell function and differentiation. And again, it's an animal that lacks a lot of commercial reagents. There's some antibodies starting to come online, which is great, but certainly not very many yet, and not all of them work that well by flow for whatever. They've been using, in situ hybridization a lot to assess gene expression. So we wanted to try and build, again, sort of a cell type ontology and see if we can detect some of the changes in the animal that we know occur in response to some sort of classical manipulations that have been done in the laboratory for several years now. So one of those is radiation. So if you irradiate these worms with 6K gamma, and then look a day later, you can see that the stem cell, which is marked by this gene called SMEDWE1, often referred to as PWE in the lab, that cell type is knocked down to essentially 0. And so if you follow these animals out, you can see that by 14 days post-radiation, the non-irradiated control animal looks normal. And then the irradiated animals are really starting to show a strong phenotype, because they've lost stem cells 2 weeks ago at this point. So now the differentiated tissue is really starting to suffer, and the worms kind of shrivel up, and eventually, of course, they'll die because they don't have the capability to, replace, the adult cells that are turning over or being damaged. And so this is well characterized, and there's transplant experiments have been done to show that you can rescue this phenotype by transplanting these PWE positive cells into irradiated animals. And so, we thought this would be a nice test case to sort of validate this type of image cytometry workflow, because we know we should be seeing a loss of stem cells fairly early, and then perhaps a loss of differentiated cells at later time points following radiation, as well as an increase in, from traditional microscopy, some folks have seen what look like these phagocytic cells that are probably involved in cleaning up dead and damaged tissues that are kind of increasing in frequency as you go to later time points following radiation. And so what we did was prepped disaggregated worm samples from non-irradiated, 2 days post-radiation and 4 or 5 days post-radiation, and ran them on the image stream. And so, again, we're looking at bright field, side scatter, and DNA. In this case, we used Herxed, a live cell assay, because there's a protocol already worked up where you can disaggregate animals, stain them with Herxed, and through sort of like a side population type of approach, sort out these stem cells for transplant. And so that assay, or that workflow already existed, so we used that to make these preps to run on the image stream. And you can see from this panel that there's a number of different cell types there with a number of different morphologies, as we'd expect, because this is an animal with all kinds of tissue types. And this was done just by gating on populations in t-SNE. You can see that each column is a population gated in our t-SNE map in FCS Express, and that by and large, we capture similar morphologies that way. And then again, we did Xshift clustering, found clusters that are statistically different between either no radiation and, in this case, late time point radiation. We can see there's several populations that are up and down between these two conditions. And then we can sort of collate all that information back together and go to FCS Express and start making some plots and layouts and kind of see what we have. So the 3 dot plots here are data that came from the dimensionality reduction that came from the Xshift environment. And so this is called a force-directed layout. You can kind of think of it like t-SNE or PCA in that the idea is to try and reduce multidimensional data to fewer dimensions. But the way these are generated is that when you run the force-directed layout, every event that's similar to another event is sort of attracted to it, and events that are dissimilar in multidimensional space are repelled. So you run this thing, and you can see that it sort of converges after a while, and then you have density regions of populations that should be similar in terms of their multidimensionality parameters, which in our case should represent morphology features. And it's hard to see because these aren't density plots, but there are regions of density here that correspond to populations. What I've done instead is color these by clusters that were statistically significant when looking from the modeling analysis in R. And I would just point out, I guess, a couple of things. One of which is if you look at the no radiation sample, this population cluster ID 5710, it's like a dark royal blue kind of at the bottom right. You can see that that's gone, almost completely gone at 2 days and still gone at 5 days. And so, our candidate for the stem cells is a population that looks like that, has a decreased frequency beginning early after radiation treatment. And when we look at a data grid of some cells that are in cluster 5710 from the no radiation sample, these have a morphology consistent with what we already know that the stem cell looks like, and we call them neoblasts in this field. They look very much like a lymphocyte. They're a small round cell with a high nuclear to cytoplasmic ratio. And then cluster 5710 is a cell that increases in frequency as you go post-radiation. And this is a slightly larger, more granulated cell. Some of these kind of have vacuoles. I only showed a few examples, but if you look through more of these, you can see some vacuole type things that might be indicative of their function as a phagocytic cell that's sort of cleaning up apoptosis, et cetera. So just from sort of this small little experiment here, it looks like we can validate that, yeah, we can detect some of the changes that we already know occur in this system, from the radiation, permutation of, it's done on the bench to try and knock down stem cells. And furthermore, we've really identified, I think, a few what seem to be pretty distinct cell type morphologies that now we can start to go back and look at the sort of nascent catalog of antibodies that some of the independent investigator labs have generated for these animals and see if any of these guys stain with antibodies that we know, for example, stains the gut epithelium or pharyngeal cells, and try to build out that ontology of cell types to help just sort of build a better understanding in the field of what cell types are here, and even evaluate some of the different dissociation preps and try and understand if we use this protease versus that protease, do we see a big shift in sort of the number of clusters that represent discrete cell types that we get out, or do we lose some cell type of interest because we used a protease that was too harsh or something? And so with that, I'll wrap it up. All of these were group efforts. I want to thank Nick Roner's lab and especially Rob Poyce. He's the project lead on the K-fish immunology stuff. In our lab, my boss, Jeff Haugs, the core head, he allows us all in the lab to really get into interesting stuff like that. And so I appreciate that and his help and support. June Park is a senior technician in our lab who is really very experienced with tissue disaggregation in a lot of different tissue types. So she did all the planaria worm preps for this, and they just look beautiful. And then Stowers, our computational biology group, basically has mentored me as I've sort of gained competency in R and some of these things the last couple of years, and they're really excellent. We're lucky to have that type of a resource here. And then, of course, Novo and Amnis have both provided all kinds of support along the way as we've kind of learned some of the new features in FCS Express and Shobana for some of the assay development and stuff as we were trying to go off-road a little bit with the image stream and get all that working. So with that, I hope that we have a few minutes left. Maybe we take a few questions, and I thank you guys for attending and listening.
All right, thank you very much, Andrew. That was a great presentation. It's been exciting on my side to kind of finally see some of the work come together in the end. And we do have a few questions here. Let me flip over the presentation real quickly to myself before we get to those. And again, for those of you who attended today, thanks for attending. We're going to go through some of the questions here shortly. But again, a recording of this webinar will be made available. And any of your scientific and kind of methods-based questions, after the webinar, if they come up after you've been kind of thinking about things, you can email them directly to acb@stowers.org. Any of the FCS Express support and questions that you may have can be directed to support@denovosoftware.com. And any of the image stream Amnis support and questions can be directed to amnissupport@emdmillipore.com. And if you wanted to kind of get started with some of your analysis in FCS Express today, we found it outlined here at the bottom of the slide, where you can go to our website to actually download the software and give it a try. But with that, we have had some questions come through, and we'll start going through that.